Hi, good afternoon. Let's get started. Before starting this session, I would like to announce there is a special issue of Geosciences designated to this topic, okay? Please, if you have paper or something you want to submit, please consider that special issue. Today we're going to start with an invited talk by Chris Williams, synthesis on the disturbance management impact on forest carbon in the United States.
Is there a pointer? Yeah. Thank you, Leo and Jingfeng, for inviting me to present here today. It's a real pleasure. I'm going to attempt, you might want to start this. Yeah, thanks. I'm going to attempt a somewhat ambitious and comprehensive synthesis of a variety of different research papers and topics that we've been working on for a little while in my group. And before I get too far along, I want to acknowledge especially Bardan Gimiri. Bardan, are you here? I think he's still getting a coffee and speaking with people outside, but he's around. A lot of this is his work, part of his dissertation research that he's just recently completed at Clark University, and he deserves a lot of credit. Other collaborators include Christopher Schwalm, who's now at Northern Arizona University. He'll be presenting some of his drought-related work on Friday at, I think, 6 o'clock or 5:15 or something like that. So I'm sure many people will be there. And if you won't have had a chance to see that, consider looking up his paper in Nature Geoscience from this year. And I'd also like to recognize Jim Kolotz and Jeff Masick for their continuing support, insight, lots of ideas for how this work should continue and progress. Okay, by way of motivation, we know that there can be large carbon legacies associated with disturbances, but we also know that these legacies depend a lot on disturbance type, disturbance severity. They can vary by ecoregion and forest type. They can vary a lot with soil types. Some of that is included in our work as well. This presents a significant challenge to quantifying the regional and global carbon balance because there's an awful lot of complexity associated with all these sources of variation. This study is, as I said, attempting a detailed, comprehensive, comparative analysis and synthesis across some major disturbance types affecting our focus region, which is the western United States and forests of the western U.S. So our approach, if you saw my talk yesterday, this is kind of a repeat slide of what I presented in greater detail yesterday with respect to fire. This is a fairly generic description of what happens in our model system after disturbances. So first, biomass is killed. We have to figure out how much, and it depends a lot on disturbance type and severity, but we know something about that. Carbon can be promptly removed by combustion or harvest, but a lot of the disturbance-killed biomass is left to slowly decompose and is emitted gradually by heterotrophic decomposition. We've had to add to our modeling scheme a delay of release associated with snags, particularly from severe fires that may kill trees and leave them standing, as well as in the bark beetle case of tree kill. There's clearly a temporary reduction in NPP in a lot of these situations, and we make some assumptions about how that's proportional to the magnitude of disturbances, in particular the severity of disturbance on a site. And we also have to then delay the allocation to wood because what comes back may involve some woody components, but often we see an increase in productivity, which is partly resulting from herbaceous or non-woody vegetation recovery. We then combine some, what I'll show, characteristic trajectories that attempt to represent the diversity of those processes across a range of forest types and disturbance types and so forth, with an area that has been affected by each of these disturbance agents at different times in the past. So we try to capture disturbance type, severity, ecoregion variations, forest type variations, as well as consideration of when disturbances have occurred in the recent past. So just to give you a quick overview of one of what I think is the key unique features of our work is that we use the forest inventory and analysis data, the FIA plot scale data, to characterize the rate of forest stock recovery following disturbances. And this is an illustration from one site. The red dots indicate the range of variation in biomass, above ground biomass in this case, with stand age according to the FIA database. This is for Douglas fir. I forget if that's high or low productivity class, but that's actually another source of variation that's important to capture. And you see some black lines going through there. We force our modeling framework to go through those lines in an optimal way by fitting net primary productivity, allocation to wood, as well as the mortality or turnover time and shedding rate associated with these forests. And all of that influences the shape and the ultimate magnitude of this over a long period of time. Simultaneously in our modeling framework then, that allows us to derive net ecosystem productivity. The derivative of the left curve, the curve on the left side, gives us net primary productivity to some degree, but we also use the model then to represent heterotrophic respiration. So we have a family of NEP curves that are associated with this particular forest type and its change over time. This is work published in Global Biogeochemical Cycles of this year, so you can see a lot more detail than I have time to present today. But this just gives a flavor for the range of variation that we can represent using the strength of this forest inventory and analysis database. On the left, we have the Pacific Northwest. On the right, the Rocky Mountain South region of the Forest Service domain. The various colors within each plot represent different forest types. The solid lines represent high productivity site classes, and the dashed lines represent low productivity site classes. You can see that net ecosystem productivity varies a lot by ecoregion, it varies by forest type, it varies by site productivity class within those. So there's an awful lot of detail that our model would have been entirely ignorant of if we weren't utilizing the Forest Inventory and Analysis database to characterize the stock change over time and the corresponding impacts on carbon balance of the landscape. For fires, we have to do things a little bit differently. In this case, now we need to consider partial disturbances, in particular associated with the severity of fires. And one of Bardon's dissertation chapters was to examine from the literature how much carbon is emitted, how many trees are killed, varying with forest types, with fire severities, and I talked about this yesterday, so I won't go through the full detail, but this just illustrates, again, we stuck with PNW, Douglas fir, to represent on top the above-ground forest stock and how it changes over time associated with a low, medium, and high severity disturbance. We use these classes because the MTBS dataset that I'll show in just a minute characterizes the area of forest that's burned annually in low, medium, and high severity classes. So we can combine these to represent the landscape scale impacts. On the bottom, we have the corresponding net ecosystem productivity curve. And again, we have these snag pools that delay the release. We also clearly see that a high severity fire kills more of the biomass, so there's got to be a larger regrowth sink associated with the legacy of a high severity compared to a more modest severity fire. So there's an awful lot of detail embedded within all of this. So we're going to take things like this, these characteristic trajectories with all those sources of variation. Oh, you can see the large emissions, and paint the landscape. We paint the landscape based on knowledge of the recent history of fires in different severities. The severities are not shown here. What's shown in the different color classes are for the blue, 1984 to, well, I can't even read it myself, different eras in recent time of fires that this record tells us about. The MTBS database is utilizing Landsat remote sensing, surface reflectance, before and after fires to map the severity and perimeters and area burned annually by large wildfires, which are the big players when it comes to carbon in these ecosystems. In the case of bark beetles, we have still another problem. It's kind of like the issue of severity, but now we need to characterize an even wider range of potential carbon consequences. The way we decided to address this is to impose in the model 1,400 different levels of tree mortality for every forest type, for every productivity class, and across these different regions. So this gives us a very detailed family of potential curves, which in a sense can be used as a kind of a lookup table. We apply these as representative of what happens when the landscape fires off with a certain number of trees killed, and that occurs in different forest regions and across these forest types. So this paper is now in review. We'll see what comes of it. But if you were here for the previous session, one of the differences that we have with some of the past work is that we're trying to translate this biomass mortality into the net carbon balance of the landscape to try and make predictions regarding fluxes as well as the regrowth that follows or that's ensuing after bark beetle outbreaks. The area that we use is from the Aerial Detection Survey database. This is sketch maps based on people in planes flying quickly across the landscape. I'm not sure if you can see it at all, but the pointer's not going to work for me. Yeah. This polygon, just within the side of that, there's a slight orange color. This is what an aerial detection technician would see, and they might put a circle around that region. And they'd estimate from the plane at a great distance the number of trees that were killed associated with that, looking at the crown damage. There are some challenges in working with this data set. One of them for us was the fact that we don't need the number of trees killed, but we need to know how much biomass, what fraction of the pre-disturbance biomass was actually killed, so that we can use one of our 1,400 curve types to look up the carbon impacts. Another problem is that the way we get around that is from FIA data we know the typical biomass stock in a forest of this type, and we can from the simple product estimate if you have a certain number of trees killed and you know the typical biomass of those trees, we know the biomass that was killed by these bark beetle events. But there tend to be significant underestimates in these data sets regarding the severity and magnitude of the disturbances, particularly the number of individuals that are killed, and so we have to deal with that. And I don't have time to go into the full detail, I wish I did, but we basically bracket the possible. So to put this into a table where we attempt to compare these different sources of disturbance or disturbance agents affecting forests of the western U.S., across these different pieces of the puzzle, we see the area of the western U.S. that's disturbed by beetles, fires, and harvest. One thing that pops out is, boy, beetles, according to the ADS data set, are really affecting a massive area. But I think Jeff Higgy would tell us, appropriately so, that some of that's a relatively diffuse effect, and so this may be an overestimate. While the number of individuals killed might be an underestimate, the area could well be an overestimate because not all the individuals in those red polygons that I illustrated are actually experiencing mortality. And just to quickly move on, we can translate these with our methods to the carbon impacts, the regional carbon balance impacts. And I'm expressing it here as the carbon disposition or change due to disturbance kill, including the combustion as well as the live-to-dead transfers and any removals across these different disturbance agents. I was frantically taking notes from Jeff's presentation because he showed something similar. I think I know why there are some differences in our numbers, and I won't have time to go into them, but they're actually comparable of similar magnitude. One of the things that pops out is that each of these have a significant effect. Fires, beetles, and harvest are all impacting the landscape carbon balance in a significant way. And then lastly, to add drought with my last couple of minutes, I won't have time to go into it, but the turn-of-the-century drought from 2000-2004 across the western U.S. was a pronounced, severe, prolonged period of dryness, and it had a significant impact on the carbon balance of the region. This is our best estimate of the impact. We see on top gross primary productivity based on MODIS as well as an upscaled Flux net product, and we see 8 to 10% reductions in gross primary productivity. But because GPP is reduced more than ecosystem respiration in general, net ecosystem productivity has a larger percent reduction. These are significant numbers, 30 to 80 teragrams of carbon per year. I'm not sure we should rely on the minus 300 that we see from the Flux Tower network. I think I would point your attention more to the percent reductions and the consistency across these methods. And quickly, just to show how these add up together. It turns out that there are more terms to be considered, but when we consider direct emissions, fire release on average about 10 teragrams of carbon per year. They kill about 20. 5 to 15 are killed by bark beetles, and that's what we think is the range of possible. It could even be an underestimate. And just to mention, I think that's a number that's of similar magnitude to fires, so that's I think, a useful guide. What does that mean in terms of source to the atmosphere? Well, they're of similar comparable magnitude as well. 7 to 10 or 12 teragrams of carbon per year. When we add harvests, the past 25 years of harvest seem to have left to a net zero sink when we apply our trajectories to that period of disturbance. There is some biomass that's removed. We're not considering in this current carbon balance the net effect of that. That will slowly leak from the wood products pool. And there's, of course, continuing uptake associated with the continuing legacy of all of these disturbances taking place. And then when it comes to droughts, these also need to be considered. And you can see there's a question mark there because we may have considered net ecosystem productivity and its balance, but we didn't consider the mortality associated with drought events. And we've heard quite a bit about that, had some great discussions in the poster session today about that. But it's a significant number. It could overwhelm and outweigh the magnitude of some of these other disturbances because it affects such a large area. Also, for context, we can consider the magnitude of the carbon uptake associated with an age structure of the forest. And we estimate that that's about 50 teragrams of carbon per year in other work. And then there seems to be an enhanced uptake recently of about 50 teragrams of carbon per year. Again, this is from our published work in Global Biogeochemical Cycles, and I don't have a chance to go into all the details, but I think this is one of the more comprehensive and detailed and quantitative estimates of how all of this is unfolding on the landscape. I don't have time to talk about all the trends, but there are very significant trends in every single one of these disturbance agents, and in most cases the news is more emission or release of carbon and reduced net primary productivity. So to jump to conclusions, I'll leave them here. Again, I want to thank Bardan Gamiri for his hard work and excellent computations to help us with this work, and I have 30 seconds for questions. Thank you.
Let's have one quick question. Okay.
Do you have any idea if there was any overlap in terms of harvest and fire? I guess by example, meaning gradient, the fine fire trees and those harvested as well. Do you have any way you might tease out any overlap between them?
So I think you're asking about salvage logging post-fire. We have not addressed that, and there is potential overlap as well as interactions among these disturbances. That's not something we've taken a look at, and so it's a gap in our group's understanding. It could be significant. I don't know. It's a good point.
Thank you, Chris. Our next presentation is given by Irene Berryman. It's going to talk about her research in the southwest region.
Well, first I'd like to thank the session conveners. It's always nice to have an abstract accepted for a talk, and thank you all for joining us this afternoon. So instead of talking about how to quantify the carbon that might be lost as a result of a mass mortality event, I'm really going to focus here on some of the variables that might control the response of a very large carbon flux to a mortality event. And that is soil CO2 efflux, or soil respiration, as I'm likely to call it throughout this talk. And I'd like to acknowledge my co-authors, my former PhD advisor, John Marshall. This stemmed from my PhD work at University of Idaho, Los Alamos National Lab, Tom Rahn, Marcy Litvak. I'd like to plug her talk Friday morning in room 2006, and she's going to present some great data from the same sites that I'm working on. At the University of New Mexico, and John Butner at the U.S. Forest Service. Oops. Okay, I'm working in a system, a pinyon juniper woodland. It's a very common semi-arid system in the Southwest that has been experiencing major mortality events over the past decade. Because of the widespread nature of this particular system, there's potentially really important carbon cycle consequences. What I'm really going to focus on here is how the limitations to this important carbon cycle flux is affected by mortality events. And so I've really identified here in this talk how mortality events, and I'm really focusing on what's going on in the litter layer underneath pinon trees. And so Pinus edulis, pinon trees, those are the ones that are dying in the system. The junipers are sticking around. litter carbon availability in the litter layer is most likely enhanced following a mortality event because of all the dead needles that fall from the dead trees. And that persists for maybe 1 to 3 years. That still remains to be seen in this particular experiment. Nitrogen availability can also be very important for heterotrophic respiration that's going on in the litter layer. And mortality may impact nitrogen availability as well as carbon availability. There's been some studies in mortality-affected pine systems that have shown a lower C to N ratio of the litter layer following mortality. And so if the C to N ratio is lower in the litter layer, then we might have an enhancement of nitrogen availability. Now, whether or not that's going to affect respiration really depends on what is truly limiting heterotrophic respiration in this system. And I'm going to talk a little bit about moisture availability here because we tend to put dry land ecosystems into this water-limited category. And that's really because ecosystem processes such as respiration do respond very highly to water events. This data that I'm showing here was collected from underneath pinon canopies. So it's heterotrophic soil respiration happening underneath pinon canopies from a mortality-impacted area indicated by the open symbols and a non-mortality affected area indicated by the closed symbols. And so one thing you notice right away is that the respiration, this is a couple years after mortality, was higher at the mortality impacted areas. And the responses to the rain events, as indicated by the yellow arrows, were much, much higher at the mortality impacted areas. And this is underneath the pinon canopies. And so what I really want to focus on is trying to answer the question, why is the respiration pulsing more in those mortality impacted areas after a rainfall event? So I think that it has something to do with how the suite of variables affecting heterotrophic respiration interact with each other to influence the CO2 efflux rate, carbon substrate. What I'm going to focus on here is carbon substrate, moisture availability, and nutrient availability in the form of nitrogen. I'm not going to go into any temperature data today. We're still kind of trying to figure out what's going on there, but the focus of my talk is how either carbon substrate or nutrient availability can really influence how respiration responds to rain events following mortality. And so first, I'm going to look for this general evidence of a general trade-off between limitations. So what I'm going for here is that the higher response of respiration to water indicates that respiration would be more water limited. And I think that as water limitation increases following mortality, that something else must be decreasing. So I'm going to look at whether it's a decrease in carbon, labile carbon limitation for respiration, or decreasing nitrogen limitation by those mechanisms that I discussed earlier. And this is a way to conceptualize these limitations. So again, I'm thinking that in the mortality-affected site that we are seeing a higher response of respiration to water events because carbon limitation is lower. Oops. Or perhaps it's because nitrogen limitation is lower in those sites. The flip side of this would be, of course, that you don't see as great a response of heterotrophic respiration in the litter layer to water events when either nitrogen limitation or carbon limitation is high. And so what's nice about these hypotheses is that they're really experiment ready, and so I'm able to directly test the limitations of respiration to water, carbon, and nitrogen using a field amendment experiment. And I was able to work at a large-scale girdling experiment in central New Mexico. There were 2 sites in September 2009. All the pinons above a certain dBH were killed. They were girdled and herbicided, and then 3 years later, I came in and conducted limitation tests underneath the dead pinons at the girdled site and the live ones at the reference site. A little bit more detail about my limitation tests. I was really focusing on what was limiting respiration in the litter layer, but I also wanted to do some limitation tests on the mineral soil surface because the litter layer can impact the mineral soil, of course. The limitation tests were conducted by simply applying water solutions with a syringe in the form of a 5 millimeter rainfall event, which is a very common rain event size in this system. And then my limitation, I was looking at the response of respiration to each of these amendments relative to the background rate. So for the water limitation, I was comparing the respiration rate following water application to the pre-wetting rates. For carbon limitation, I looked at the respiration rate increase following the addition of a sucrose water solution, and I compared that to the water-only treatment. And to look at nitrogen limitation, I applied a sucrose and an ammonium solution to the soil surface, and then my background rate was then compared to the rate following the labile carbon or the sucrose application. And my respiration rates, the limitations, were measured using a continuous system, the automated carbon efflux system, and their closed chambers that use an open method of calculating a respiration rate. So my simulated rainfall, just the water-only applications, revealed that there was indeed a larger response of respiration to water additions at the mortality-impacted site, as shown there by the open symbols. And so my hypothesis that respiration was more water limited at the girdled site was indeed true. And so now remember, I'm looking for trade-offs in these limitations. Now, why did I see a higher response of respiration to water at the girdled site? Am I seeing a lower carbon limitation at the girdled site compared to the reference site? Or am I seeing a reduction in nitrogen limitation? So I did find some support for decreased carbon limitation at the girdled site. You can see here that there was a trade-off between increasing water limitation of respiration at the mortality site alongside a decrease in carbon limitation. But I still wanted to know about nitrogen. Now, the litter C/N ratio was actually not different between the two sites, which was a little bit unexpected. But that would indicate that we probably wouldn't expect to see a difference in nitrogen limitation because C/N is not different. Not only was that true, but there was actually no detectable nitrogen limitation in the litter. So respiration did not respond to ammonium additions at either, or it did not increase at either site. I do want to point out, though, that at the reference site, respiration actually did decrease in response to ammonium, so that was surprising. But no, so we had evidence of carbon limitation in the litter, but not nitrogen limitation. However, in the soil, on the soil surface, and this would be the soil surface that's in direct contact with the litter layer, I did find that respiration increased in response to nitrogen additions at both sites. I did find that the nitrogen limitation was actually higher at the girdled site compared to the reference site. And I think that this is a response to all of the additional carbon that was present in the litter layer at the girdled site. I think that the additional carbon induced a nitrogen limitation as microorganisms were decomposing the labile carbon. I think that the soluble nitrogen in the soil was under higher demand, and so it was more nitrogen limited. And I also want to point out here that carbon limitation in the mineral soil was the trends with carbon limitation in the mineral soil were consistent with the trends in the litter, in that there was an increased carbon limitation in the mineral soil at the girdled site. So overall, I did find this pattern that not only did carbon and water limitations trade off in the litter layer, but also carbon and nitrogen limitations did trade off between the mortality affected site and the reference site. So as nitrogen limitation increased, carbon limitation went down, and vice versa. And bringing this back around to mortality in the carbon cycle, I think that what's going on with some of these pulses of respiration following rainfall events is that it appears that respiration is more water limited when you have a mortality situation. You have additional litter fall on the litter surface, And that is actually reducing the carbon limitation of respiration in that system. And so I think really what's going on is that mortality events are shifting the limitations for carbon fluxes, and that's something that should really be considered in carbon cycle predictions in response to mortality events. And just real briefly, I'd like to thank all the people who helped throughout the years to culminate in this talk.
We have time for a couple of questions.
Are there any evidence of changes in microbial communities or the results like this?
Yeah, so the question is, are there any evidence of changes in microbial communities in response to the mortality event? Yes, there has been some mortality or some microbial work done at the sites. And I don't have the specific data. Marcy, I don't know if you're going to be talking about any of that stuff at all. No, that would be Bob's. Yeah. Okay, I think that there have been some changes. The ones that I'm aware of are the mycorrhizal component has changed. That's all I'm aware of.
I think you mentioned that it's more water limited in the case of the early.
Yes.
Is it that it's less carbon limited?
Is it because of? So, are you asking if I can imply causation? Is that? Oh.
Is there just more carbon around to be pulsed?
Yes.
So it's less carbon limited.
Yes.
It's more water limited.
Well, the way I'm defining water limitation, it is because I'm looking for a response of respiration to a water addition, and so that's how I defined water limitation. There was a higher response of respiration to the same amount of water being added to the surface. So I don't know if I don't want to confuse it with an idea of being indicative of water availability. Is that maybe what you're thinking? Because it's not. It's a direct indication of how does respiration respond to an increase in a variable. So.
Okay, thank you.
Thank you.
Our next presentation is the invited talk from Professor Jichuan Chen from the University of Toledo. He's talking about the climatic extremes on carbon dynamics.
Well, thanks, Leo, and thanks, Jim, to put this session together. Do I have a pointer here? Oh, you have. You stole the pointer. Okay, well, what I'm going to do is that first I need to get rid of that significance because it turned out I have probably many of you that didn't conduct a statistical test. So, but I'm going to focus on how climatic extremes affect the carbon water fluxes. So I'm going to also use the 4 case studies to demonstrate where and when these extremes can affect carbon fluxes. So specifically, I'll use the long-term data at opening Ameriflux site, and also then the second case is going to be a cluster of tower that they are located in the same watershed, so that where the landscape, soil, vegetation are the same. And the third one is trying to use another cluster tower site where we look at it, how different bioenergy system located at a KBS, the same soil, same of everything, a climate, and except land use history. And lastly, to use the Mongolia Plateau project to illustrate at how extreme events may happen in space. So first, let me bring this the opening flux tower. The results are from ASCO Norma's paper back to 2004 or 2008. Basically, he reported that in the 2 years, 2004, 2005, if you look at these numbers, the NEP values, because of this drought, can be reduced in those 2 adjacent years by 40%. And what's interesting is that now we have the data to show here I included only 60 years, 6 or 7 years, but the annual NEP value can be as low as 1.9 tons, but can also be as high as over close to 6 tons. So there is a lot of intra-annual variabilities, which we know. There is no surprise for this intraannual variability. What here we're trying to do is to ask a simple question. What are the contributions of these extreme events during the course of a year that helped either reduce or increase elevated NEP values? So here I use one specific example. If you look at the the daily carbon fluxes NEE value of, I think that's 2004. Specifically, I want you to pay attention to these two pulses. That's the two pulses we got raining in the summer of the Northwest Ohio. Suddenly we got a week-long cloudy, raining days, and then those weather underturned the ecosystem from major carbon sink to a carbon source. This is like in June and July. So forest in the summertime can be a carbon source. Not always true. But more importantly, I want you pay attention to these numbers. That two events, extreme events alone, counted roughly about 9% versus about 20% of the reduction in EE. Just a 2 year. It was a 2 weeks period, and the affected 30% of the annual NEP. So that's one significant. So now we're in the process of finishing this systematic analysis of the fluxes over the last 8 years. So we're using these anomaly issues, basically look at how this temperature, moisture, all these climatic drivers are different from the long-term means. For example, here we use these 2 standard deviation of anomaly issues. And for example, you see obviously that's one of anomaly because the standard deviation, the anomaly index exceeded 1.5. But we all sometimes at the temperature heat waves and the drought, they don't always show up together, right? So we have multiple variables. So we have to look at the extreme events of all the variables, whether it's the air temperature, soil temperature, or VPD, or it's because of soil water content. So we have completed these all climatic variable drivers. I'm not going to show every year, but here I just want to share with you one table of how all this, altogether we identified about 28 events, climatic extremes over the past, from until 2010, I think, yeah, from 2004 to 2010. Here are some numbers, okay? Here are the NEE value for the monthly NEE values, but here are the percentage. I just want you to pay attention to this percentage of those extreme events affected. These are the relative value, the absolute values. These are the percentage of relative values. What is clear is that is extreme. First, that extreme events can cause whether NEE, ER, ecosystem restoration, or GEP, either positively or negatively, but the percentage first. Also, we know the NEE is the difference between ER and the GEP, and sometimes it can be the opposite, sometimes affect the same with same directions. So, but overall, you can see that the effect on the GEP is way more than the ecosystem restoration. These slides present a wavelet transform of temperature and carbon fluxes. This is a complicated slides, but what I want you to pay specifically is 2007. That's one of the summer we have a lot of the heat, drought, and that's a really different. Every summer we have a little bit of this, okay? These are some heat waves, but this year we got a lot. And surprisingly, but expected, here is the wavelet transform of NEE, carbon fluxes. You see that exact pattern. So there is a direct. We may not see 100% correlation between temperature and NEP, but here from the wavelet, that pattern, if you look at this pattern of the heat waves versus, let me go back to here, temperature and carbon, almost identical. Okay, I want you to pay to the shape of these wavelet transforms. So this is like ignoring this extreme is a mistake. So second case study I want to present to you is that, okay, now that's one side. What happens? How different the ecosystem, where, for example, in the Maumee 1 watershed, we have several flux towers, so we can compare. Here is virtually the same soil and the same climate. So how the same extreme events can affect a different ecosystem. So here we have like a prairie system, cropland, wetland, and forest ecosystem. What are the differences? So Hosen he has a poster tomorrow, or it's already done. Basically, look at this year. 2012 is extreme year, right? We all know that. Warm winter, strange March, April in North America. So you see these kind of temperature responses, but that's expected. But what Hosen is trying to show in this paper is I want you to look at is both NEE and ET values. And the cropland here virtually shows very little effects on the ET, but the carbon flux is yes. And also there's a spring drought versus the warm spring that produce the opposite effects. Versus this is the marshland versus the oak company, that's a forest. Forest, again, is 2011 produced lower NEE, but marshland actually the opposite ways. So the same extreme events can produce opposite or no effects, depends on which variable we're talking about. And Hosen also look at it, okay, we're looking about physical models. Here we know Q10 models or some of the light response curves. These are the slope and maximum photosynthesis. Basically, marshland, cropland, oak openings, we know this extreme also affects these coefficients involved in these parameter biophysical models greatly, but also depends on which ecosystem. For example, cropland, for the reference respiration, didn't show effect too much. And the next case study I want to quickly go over is look at KBS. Many of you probably know KBS is a flat area. It's a homogeneous landscape with the same climate. Here we have a 3 bioenergy system, switchgrass, corns, and native prairies. The only difference is these bioenergy systems were created from either CRP, conservation reserve prairie, or the cornfield, agricultural field that has been managed in the past 20 years. And so in order to reduce the system differences or the differences among these bioenergy systems, obviously we have 3 bioenergy systems and 2 land use history, right? So we have full factorial design for all of them. So back to 2009, we plowed all the fields, regardless of land use history, plowed the fields, planted the soybeans to homogenize all 6 sites. And here we follow the changes of carbon. From the very beginning, we followed carbon, water, and energy fluxes over the past 3 or 4 years. So right now we have a corn, switchgrass, and then prairie, also the same thing from those sites converted from the corn field. So I'm just going to only share 2 slides. Here is the change of the NEP, GPP, or ecosystem restoration at 2 sites. Both are the top one is converted from CRP, or we call it prairie ecosystem, and the bottom one is converted from the cropland. So obviously, let's say before the conversion, here's the conversion period. Well, we know native prairie already has much higher NEP values or GPP and lower, also higher ecosystem restoration. Cornfields here, as expected, after 20 years, it's pretty much carbon neutral. But what's interesting is that it depends on which landscape. Now I want you to pay attention to the difference. Now, last year, look at the differences. It does not mean where we convert the land from native prairies will produce the higher NEP, which is virtually similar to the biofuel production. So that's the change at a switchgrass system. And the next thing is from the conversion from the cropland. Again, it's so different. Initially, they are the same, but now, in recent years, they're almost the same. And they are significantly different, both in terms of the GPP respiration or NEP native. So last case study I want to share with you is let's look at the space. Those are some of the time in different climate. Now here I want to move you to the Mongolia Plateau. We know Mongolia Plateau is in high latitude with base elevation of almost 1,000 meters above the ground, but it's also the high latitude. of 40 degrees or higher. And IPCC clearly showed that its part is one of the hot spots in the global scale. So here we deployed a cluster of, for example, mobile flux towers and permanent ones combined with other biometric measurements, bind models. Well, there's no surprise here. On the Mongolia Plateau, for example, number of the cold nights versus warm nights all decrease or increase, and depends. They're just really the same pattern as IPCC predict for any other region. And also, this place, also unlike any other regions, there is a clearer temperature from north to south to north. You have the expected temperature gradients, or from southeast to northwest, the precipitation gradients. But what's a surprise is when we look at the climatic change, let's say decadal slope, change of the temperature or precipitation, right? That's what we call the change, the rate of the change. Again, the slope over the year. Here are the two maps produced by Ranjit and John here. So basically, the temperature-wise, we identify the cool spots or versus the hot spots on Mongolia Plateau. We know specifically, for example, here, global warming is not happening. It's getting cooler. And of course, precipitation varies also. But what's the surprise is that when we have those hot spots, with a cool spot or hot spot of change, it doesn't reflect at, for example, GPP. So there is no direct coupled. So what this suggests to us is we have to place all this into a tightly mechanism-based processes. So again, here are the conclusions. I'm not going to read. You can read all this, and I won't take the 20 seconds to answer any questions you may have. Thank you.
One quick question.
Yeah, Marcos. That's the mode. Mode is the GPP. Yep.
Okay, that's quick. Thank you.
Okay, thanks.
Our next talk is kind of different. It's given by Biamang Yu. It's going to talk about hurricane impact on DOC exported in streams and rivers, I think.
Well, good afternoon, everyone. My name is Brian, and I'm really glad to be here, and I want to thank the conveners for letting me come here and talk. And today I'll talk about the dissolved organic matter export from a forest to watershed during Hurricane Irene. And somehow I was lucky or stupid enough to be in the middle of Hurricane Irene when it came to Cascade Mountains. And there was a site that I was using for my master's research, and today's talk will be based on some of the data that I collected during my master's research. So before we talk about hurricane, let's talk about some of the background information so we can have a better grasp of the topic and also what it means for the global carbon cycle. So one thing that we are noting, one thing that we noticed about the carbon or the climate in the past is that we are seeing more precipitation and more frequency of intense precipitation. And this is not only true for U.S., but also there are many regions in the world where we're seeing a similar effect. And we also experienced hurricane intensity change in the eastern U.S. and Hurricane Irene, Lee, and Sandy are the good examples that is coming to play in our environment right now. And what is more alarming, as we heard in the first session, is that there's going to be more Category 4 and 5 hurricanes in the future. So the question that we come to naturally ask is that what is that going to mean for the carbon cycle? Well, the part of the carbon cycle that I want to talk about is somewhat different from the systems that we were dealing with before. I'm going to focus on inland water systems, and these inland water systems, streams and rivers and lakes, they used to be considered as a passive conduit, just a pipe that transports carbon from land to sea, but now we come to realize that this is not a passive system, but it's a very dynamic system that moves carbon, that stores carbon in the bottom of their system, also emits carbon to the atmosphere, and whatever is remaining at the end of the system, it is what gets transported to the coastal areas. And the latest figure of that carbon evasion from streams, which we just got on Monday here at AGU, is 2.4 petagrams per year. And if you compare to the anthropogenic signal of CO2, you can understand that this is not an insignificant flux. And within those fluxes, what I study is, or what I'm going to focus today is disorganic carbon, or DOC. And this is the global estimate that we have. And keep in mind that this is a riverine carbon flux to the ocean, and this is only the remainder of whatever happened during that transport. So it is not only significant in terms of quantification of the flux, but it also is connected to a lot of things that we need to relate to. For example, DOC is really critically important for microbial metabolism or and biological pumping in the ocean. It's a source of carbon dioxide evasion from the stream, and also it will form disinfection byproduct in water once you try to clean the water for drinking water purposes. So it is not only a significant flux in terms of numbers, but it also has a significance because it is really connected to a lot of ecosystem services that we rely on. So then the question is, when does DOC get exported? Well, it turns out that in most climates that we are dealing with, DOC gets exported when you have a higher discharge. And it's pretty obvious that when you have more rain, more discharge, DOC gets washed from the land and it gets transported to streams and rivers and it gets transported. But the question, for example, 86% of DOC is exported during events or precipitation events in Northeast, and more than half gets transported during intense events. And the question that I will address today is, what happens when you follow this curve and go all the way up here, when you have an experience or when you have an event that is unprecedented and that is totally extreme, and that is where Hurricane Irene comes into play. So Hurricane Irene, as some of you can remember, was one of the most deadliest hurricanes that we've seen in Eastern Coast. It affected almost every single state on the eastern border, and it produced record-breaking precipitation throughout its travel. And my site, noted as a star, is right in the middle of the Catskill Mountains, and you can see how a hurricane is moving up, and the star is right in the middle. And zooming into the site, this is my site, Esopus Creek. It's in the heart of Catskill Mountains, and it is mostly protected because it is a primary water source for New York City. And because of it's protected and because it is protected by state and the city, there has been a pretty good record of data on discharge and also chemical data. And this site is, as you can see, is mostly forested, and it's about 98% forested. So it is really rare place to study in northeastern U.S. where you have a good record, but you also have a large protected watershed. And for my master's research, I was going out there whenever it rains or whenever there's a big event to just take samples. And I had collected about 114 samples during 2011, which some of which include base flow samples. And there's been some monthly samples from USGS from the 90s, and I'm using all those data today. And as I said, I was stupid enough to be out in the field when Hurricane Irene was coming. And this is a site that I was at, and this is a nice-looking second-order stream, and that's the USGS gauging station that I use for my sampling. So how big was Hurricane Irene for the specific watershed? Well, it produced about 30 centimeters of rain in just 3 days, and I think it's actually an underestimation because the gauging station did not work for one of the 3 days. And just the precipitation by itself, it was a 200-year event, and there was a 330-fold increase in about 9 hours with the onset of the storm. So the water discharge increased from 2.5 to about 800 in just a few hours. And you can see from the pictures here how damaging the storm was for the watershed. So let's start, talking some numbers. What really happened? Well, no, one more example is this is a watershed that's right next door to my watershed, and they have a longer historic record that goes back to 1904, and you can see how Hurricane Irene really stands out even in that 100-year record. So this is a discharge that you can see in the background. Notice that the y-axis is in log scale, and these are the samples that I got during Hurricane Irene, and you can see the rapid change of color in the samples that I have. And DOC concentration, of course, as I expected, it was controlled by discharge increase. But what is really surprising here is that there is no dilution effect taking place. So people used to think that once you go past a certain threshold in discharge, there will be a dilution because there's too much water and there's a limit to DOC that can be leached from the watershed. But that was not the case here. DOC just skyrocketed. And DON or dissolved organic nitrogen was the same case. It just went all the way up to 0.25 milligram per liter. And these two high concentrations that I saw, the third one here and the third sample here, they were both the highest concentration of DOC and DO1 in recording history for that watershed. So then the question we need to ask is, well, how much was the total? Well, using this semi-high resolution data that I have and also the 15-minute discharge data from USGS, we can calculate that the amount of export was about 100 tons of carbon and 5 tons of nitrogen. And this is a really small number of carbon once, when you're used to bigger scale, but this translates to about 43% of the annual carbon export from the specific watershed and also 30% of nitrogen export in that watershed. So, and if you think about it, it was only 5 days of the year and exported 43% of its annual total output. And another question that we need to address is what kind of carbon got transported, because that is directly linked to the ecosystem processes that happen with the receiving water bodies. So using specific UV absorbance, which is an index for aromaticity of the carbon, we saw that there's a higher aromaticity in the carbon that's coming off during the peak flow, indicating there's more humic material that's coming from the land. And also using excitation emission matrix, which uses the fluorescence to characterize the composition of the DOM that's getting transported, we can notice that there is not much change in the composition. There's not a lot of protein-like DOM that's coming, but it's mostly intensification of terrigenous humic material. But if you go further into the difference between humic materials, you can actually see that there is a more increased export of reduced humic materials. So there's a flow path change in the land that brings humic materials that was not exposed to flow before. And I also performed lability tests to see how reactive this DOC is once it makes to the receiving water bodies. And surprisingly, it was not that labile at all. I mean, if you look at the third sample here, which was collected with the peak DOC concentration, its lability is almost down to 2 or 5. But the thing that we have to keep in mind is that the lability actually goes up once you pass the peak of the hydrograph. And even at 10% of lability, we are exporting a lot of carbon because the discharge is so massive at this point. So why should we care about this big flux with reactive carbon? Well, one thing that we need to keep in mind is that this is a huge event that exported 46%. That is a very unprecedented export in aquatic biogeochemistry. And this also shows that there's no limitation to DOC export. So it's not DOC limited, but it's a discharge limited system where DOC can get transported if you just put more water into the system. And also, I mentioned that there's an export of labile carbon, and when you have labile carbon and also UV or the photoreactive carbon going downstream, you can also interfere with the microbiome metabolism or the UV processes that can control the water quality or the ecosystem status of the receiving water bodies. And there have been many cases for example, in Thailand, in Taiwan, where intense monsoon precipitation can overturn a status of a small lakes from autotrophic system to heterotrophic systems. But because this was not a research that I planned ahead, I could not submit a proposal to NSF saying, I want to study the hurricane in the middle of the hurricane, I want to study the 500-year hurricane. But because we couldn't really handle it, we couldn't really estimate where all these changes will occur. But we actually could find some signs of change that is happening in the Hudson River. So this is further downstream in the Hudson River, and this is the data from the HRESCO. And what's happening here is that as Hurricane Irene is coming down Hudson River here, indicated by turbidity in red dots, you will see a huge drop in oxygen. And Stuart Finney said that this is a Hudson River taking a breath, and that's exactly what's happening here. And the negative effect was not so bad because the water that's coming during Hurricane Irene was highly oxygenated. And also there was an elevated bacterial growth in the river itself for more than a month after Hurricane Irene. So another thing that we need to address is the water quality. And as I mentioned, there's a high SUVA values indicated during the hydrograph when you have a peak DOC concentration. While those samples also had highest DOC concentration, DON concentration, but also high SUVA values that I've recorded. And high SUVA, if you are having a higher SUVA in a given watershed, it usually indicates to a higher formation of disinfection byproduct or DBP. And this is a carcinogen that we find in water, and it already is the biggest concern for drinking water quality across the country for big cities. But one thing that I need to tell you is that New York City knew about this, and they could actually control the formation of DBP. So New York City's drinking water quality was fine after Hurricane Irene. And this is some of the picture that shows the physical damage of Hurricane Irene. Down in the Hudson River, they saw greatly reduced submerged aquatic vegetation, and they did not recover until the summer of 2012. And they usually see recovery of SAVs pretty fast, but they are still studying this, but they will see an increased impact or continued impact of Hurricane Irene in the main Hudson River. And last thing that I want to touch on is how important is going out to the field and sampling during extreme event like this. Well, it turns out if I was not out there and use everything else but the data that I collected, I would actually have underestimated the flux by 40%. And if I change the way I use my load S model, which is the USGS standard model for calculating load, and change the way where I use every sub-daily values to increase the accuracy of the model, I still underestimate by 16%. So this tells you that you really need to have a representation of extreme event if you want to model or calculate the flux during those extreme events. So going forward out to the bigger scale, I mean, what does this mean for us when I mentioned there's an increase in hurricane intensity, and we had a session about changing hurricane intensity in the first session too, but this means that we are going to have more export of terrestrial carbon that's going farther downstream, and also this is going to be more reactive organic matter. So what this means is that there's a potential increase in the strengthening of land and sea linkage in carbon cycle. More carbon is coming from the land, and more will be reactive, or more reactive carbon will be processed in inland systems or inland water systems, and more will get transported to coastal areas, which means that these fluxes that we are still having a hard time understanding are all going to change because hurricanes gradually. Well, so the take-home point of my presentation, well, Hurricane Irene exported a lot of carbon, and export of such magnitude could affect the aquatic metabolism, UV processes, and water quality. And as I mentioned, sampling during peak flow is really important for accurate estimation of extreme event fluxes and increasing intensity of hurricane, and also, increasing intensity of extreme events will potentially strengthen the land linkage of carbon cycle. And with that, I want to thank all the people who helped out and people who came out to my side and enjoyed the beer with me. And I will take your questions. Yes.
Much of your talk focused on downstream effects. I'm just curious, do you have any comment or speculation on any upstream effects in the watershed? Do you suspect that all of that carbon nitrogen being basically washed away, would that affect productivity of the watershed itself?
Yes, so I did a back of the envelope calculation to see how much we actually lost from the watershed itself. And the DON, for example, was about 0.5% of the soil reservoir. So if you consider like 1 or 2% of the nitrogen or carbon gets sequestrated in soil, it really is a lot of carbon. And also for receiving water body of the Ashokan reservoir, the carbon flux that came during Hurricane Irene would translate to about 50% of its standing DOC stock. So, but the good thing is, people at USGS New York, they actually studied the insects and invertebrates living in the streams, but they saw a really good recovery of all the species. So, thank you. Thank you.
We're going to go to Kansas grassland. Next talk is about climate extremes of grassland, carbon cycle, and water cycle. Thanks.
All right, thank you. I'd like to thank you all for sticking around. And today, what I'd like to talk about is how changing extreme weather distributions, primarily temperature and precipitation, what the impacts of that are on water and carbon cycling in grasslands. And over the last few years, we've been doing a lot of work in Kansas looking at what GCM predictions from the IPCC scenarios suggest will be likely climate change impacts in the region. We're seeing probably temperature changes just in the mean temperatures of about 4 degrees over the next century, and probably either no change in annual precipitation or a slight decrease in annual precipitation with an increase in the extreme event nature, the periodic nature of this distribution. So becoming less frequent, more intense precipitation storms. And so our real interest here was on what's happening with the carbon and water cycling, as I said. And so a lot of this work is all being done at the Konza Prairie LTER site. So it's a site near Manhattan, Kansas, and central Kansas. It's a tallgrass prairie ecosystem managed at watershed scale, with each watershed having a combination of either fire burning and or grazing by bison or cattle in various treatments and timing. And so in a normal year, we see kind of lowland grasslands. So this is an annually burned C4 grassland site. In a normal year, and around mid-July to the beginning of August, we see grasses on the order of about 2-meter height in the lowlands. So we see, a fair amount of biomass accumulating in these lowland areas. And then in a few years ago, we had the same time of year, same site, and we see that we had a large reduction in the above-ground biomass. So this was not this year, this was last year's heat wave. And so we were thinking, at this point, we were really excited that we could do this kind of look at the extreme event nature of this. We could actually say we had a strong month-long period of high temperatures, very low precip, reduced precip over this time, obvious implications for above-ground biomass. And then, of course, this year came along, and so as we were processing all of this data to look at the nature of the extreme event, we see an even larger extreme event occurring this year. So we're left with the question then of what's the impact of the timing of the magnitude of this extreme event. So if this heat wave starts around in July 1st or so and lasts about a month, but what if we had a similar magnitude event occurring a month earlier? Or what if we had large precip events occurring later in the season or earlier in the season? Do you see the same kind of dramatic implication of above-ground biomass at an annual scale? So over the last couple of years, my colleague Jesse Nipper at K-State has been accumulating every 2 weeks. He goes out and does a series of sampling, above ground biomass, clippings, temperature, soil moisture, a whole host of other things. And so this is in the same watershed, and we can see as a function of topographic position, upland, slope, or lowland area. And we can see that this magnitude between 2010, which was a more normal-ish year, to 2011, we see that this drastic reduction occurred right in that heat wave period, which is that, do I have a pointer? This yellow period here, this yellow time. So this is our month-long extreme event, and you see that this reduction in the above-ground biomass in the lowland area is drastically reduced, while the upland area is not quite so affected. And because of the timing of this event, so just comparing from an eddy covariance tower, we see that this event occurs about here, and this is about where we see divergence in the carbon fluxes measured on the eddy covariance site. So again, the question is, if this event had occurred here, would you see the same kind of drastic magnitude? Would you see reduction as this function, this timing of this year? And we have several years of eddy covariance data at this site, but we don't really have enough to look at long-term extreme events and the implications of extreme events. So we're going to turn to a modeling approach to look at this. And so as we go through this, we want to look at what are the impacts of this, and are various models actually sensitive to these timings of these events. So here's a record of a couple of years of eddy covariance data. Like I said, we don't have a long enough time record to actually look at extreme value distributions. So we're going to go to an agro BGC, which is a version of Biome BGC, which was provided by Alan DeVittorio. And so just on a couple of years of validation or calibration data sets, so some years we get fairly good agreement. Other years we seem to be missing this kind of early season spike or early season growth. But we say that since we're looking for timing, we're going to consider this good enough to proceed on looking at the timing of the event rather than necessarily the magnitude of the fluxes. And so how we're going to do this, we've developed a technique that we're calling critical climate period analysis. And this is basically by taking a day of the year of daily meteorological data, so air temperature, precipitation, something like that, and correlating it with above ground biomass at the end of the season. So for this, we're going to use the LTER data set. So we have about 30 years of above ground ANPP clippings, and we have the weather station data. So we can look basically at a R squared between a linear fit between the precipitation, say on January 1st for all years, and what's the relationship between that and above ground biomass at the end of the season. And so we see for precipitation and above ground biomass, we're seeing particularly sensitive to this kind of 180 to 200 day period on the, with a window size on the order of about a month and a half or so. We see very good agreement. And there's actually a higher correlation with this, with these values here than actually using annual precipitation. You get higher correlations between just using this particular window of precipitation. And so we look at the same thing for a temperature effect, and you can see that the temperature effect is a much shorter window, so on the order of a couple of week window, right around this time where we saw the heat waves of the last 2 years. So right around July 1st, incredibly sensitive. The annual biomass at the end of the season is incredibly sensitive to temperatures in that particular window. So then we wanted to see if the actual models can pick up on this kind of dynamic. And so when they look at the precipitation variability with annual growth, so this is the plot that you saw just before. This is the agro-BGC. See, in general, it's picking up in the right sensitivity and the right timing. It gets a little off on the actual value. It's a little overestimated, and the window is probably a little over-exaggerated, but it's generally sensitive to this. But this one isn't particularly surprising. It's saying that above-ground biomass is sensitive to growing season precipitation. Right. When we go to the temperature, where we had the actual data is very sensitive to maybe a 2 to 3 week window here, we see this model is, again, a little overly optimistic in terms of the magnitude, and it's saying, again, kind of growing season temperature. So we're a little off on this, the sensitivity of this particular window on the temperature extremes. But so then what we wanted to do is actually look at the impacts of climate change on these regions. So we can take different scenarios, and so using a stochastic weather generator, we're going to do 400-year simulations with basically altering the precipitation to make it more episodic, so larger intensity, slight reduction in annual magnitude, increasing temperature by about 4 degrees over the course of 100 years, or we can combine the reduction in precipitation with the increase in temperature. And we can look at what these effects are on various dynamics in the model. So these are plots of the box plots of the annual averages of the 10 years of 4 different realizations of 100 years. So the kind of end of the century simulations using 40 years representing the last decade of likely outcomes of the century. So here's the impact on annual soil moisture. So the control is just kind of rerunning the precipitation. You see reduction in precipitation gives you a reduction in soil moisture. Increase in temperature actually gives you an increase in soil moisture, which I'll show you why here in a second. And then the combined effect, you're actually seeing fairly similar to the control. When you go to the fluxes, you see kind of no real change in the total amounts for the carbon flux, but you do, when you start temperature only, you see kind of a reduction in the magnitude. The precipitation, you get more interannual variability. ET kind of follows the same story as kind of the soil moisture. In the control case, just a temperature, you see a slight reduction, which I'll show you why. If you decrease precip, you decrease the actual ET, and then you combine them both, you get a reduction relative to current kind of control scenarios. When we look at an average annual cycle, you increase the temperature, as part of the scenario, you increase the mean temperature, and then you decrease the precipitation. So this is just the control versus the combined effects of temperature and precip. When you go to the soil moisture here, you can start seeing what's happening. So this system is heavily reliant on winter recharge normally, and what we've seen over the last 30 years of the LTER. So we're seeing a slight shift in this cycle here. This recharge in here and the combined effects, I think, has to do with the fact that we're seeing a reduction or a shift in the growing season. And so I think that this is causing earlier senescence, which is actually reducing the ET in these periods because you have more biomass, which is shading evaporation. That's my best guess at this point. Looking at that, that's why you're seeing higher soil moisture at this point. Is basically a shading effect with senescent vegetation is limiting the evaporation at these times. So we can go to the critical climate period analysis now for all of these different scenarios. So we have for this slide, it's all with respect to annual biomass at the end of the season. So here we have just the precipitation effects on annual biomass for the control scenario, going through just the precipitation, just the temperature, or the combined effect of both scenarios. And as you can see here, as we go through the control, you're seeing it shifts from what we see in the data to a little bit more emphasis on late season precipitation, which is about the same. If you just reduce the precip, it's still sensitive to this late season precipitation. You just increase the temperature, it becomes a pretty pronounced effect. So again, this is the precipitation effect on annual biomass. It's pretty pronounced at late season precipitation. So if you do get late season precipitation, then you're kind of offsetting that senescence and that shift in the growing season. And so you're able to keep the grass alive a little bit longer, gives you a higher biomass in this late season. You do a combined effect of both the temperature and the precipitation, and the model is saying that you get this sliver here, in which you can, so just incredible sensitivity in this particular time period. For temperature, you can kind of see losing kind of any kind of a relationship here. Soil moisture, we're kind of losing this winter recharge effect when we go to the combined effect here. So you're seeing the sliver kind of develops here, which is, again, I think related to this precipitation effect and whether or not you're able to increase the GPP over the course of the season. When we look at the evaporation, so now just this is annual daily precipitation, daily temperature, or soil moisture with the annual water use. You start seeing as you start combining things, this kind of mid-season, growing season precipitation here. Temperature, you see very little effect going across the board on annual evaporation. And soil moisture, you see pretty much the same story as you saw with the carbon side. The shift from this winter recharge, high sensitivity to this winter recharge, to a particular sliver of time during the year. And so when we can think about what this means, it's, in one level it could be real, but in another level it could be used as a model diagnostic, right? These are the particular time scales that the models are sensitive to. So one thing that we have to do there is kind of start looking at comparison with a whole host of different land surface ecophysiology models to actually know, is this an artifact of sensitivity of agro-BGC to particular events at particular times of year, or is this actually indicative of what's actually happening in terms of plant physiology, in terms of the annual growing season? So yeah, I'll go ahead and stop there. And I think, again, that we just need to kind of focus on these time scales, various time scales, and looking at these, a different way of doing model diagnostics, basically, in addition to seeing what climate change impacts are.
Thank you. Well, time for one question. Okay, thanks. Our next presentation, I don't even need to look at my note. So, Babe Lloyd's going to talk about her work, very comprehensive work in Northwest Pacific Northwest. Thank you.
Okay, this is all right. Thanks for having me, and thanks for sticking around, everybody. Okay, I'm combining some of our data sets from Flux sites and various models that have been applied in our region to look at how forests have functioned under past climate and land use, how forests will change with future climate and CO2, and then how will proposed mitigation actions affect forests under future climate and CO2. So ecophysiologists were granted a really great period of time where we had a pretty extreme drought and as well as warm temperatures in the early 2000s, and this gave us a real opportunity to challenge our understanding of sensitivity to drought. And we had studies going on at Flux sites in Oregon in mesic Douglas fir and semi-arid ponderosa pine as shown in the graphs. And we also had an experiment going on in young ponderosa pine. And in general, we found that GPP, root respiration, transpiration all declined by about 40 to 50%. And the decline was very sharp after relative extractable water was below 50%. So that's kind of a number that looks like a threshold that we should be focusing on. And as mentioned before, confirms some of that. And then we had 30 years of tree ring data that we looked at both sites to see what the longer term effects were. And we compared that or looked at the correlations with Palmer Drought Severity Index. And we saw that in general at both sites that there were carryover effects on growth for 1 to 3 years after a significant drought. And then, as you've seen and heard earlier, there's expected more drought-induced stress and mortality in the future. So we've got to get our act together on this. Next, just looking at response to fire and harvest. And many of you might be aware of the Pacific Northwest Forest Plan that went into effect in about the 1980s, and that's when the intent was really to protect habitat of spotted owl. But what it did was it reduced harvest by about 80% on federal lands, and there was no change on public or private lands. So in the graph there, the upper graph is private lands, and lower graph is public lands. And the light bars are net ecosystem carbon balance. So that's net ecosystem production minus fire emissions and minus harvest removals. And you can see that on private lands, they were near carbon neutral for net biome production, and public lands, it increased. So harvest had a huge effect on the land-based carbon sink. So the next question is what will happen to carbon balance in the future when we have vulnerability and mortality may increase. Well, Nicholas Coops led some work in the region. This was a NASA-funded project where he was looking at species richness, and it was based on the 3PG model Dick Waring and Joe Landsberg had developed, and I worked on in my early years. And that model is physiologically based, and then he did a hybrid approach and brought in decision tools for deciding when a system might become more impacted, such as reduced soil water content by half. So this model has modifiers that are VPD, drought, and temperature. And the example is one of the modifiers is reduced by 50% and over a number of years, and you get an idea of what might be there in the future. So here's current, and then I'm going to show other decades into the future, so 3 different ones, and you can see where the shifts are. It's basically a loss of species richness is predicted for the coastal areas, and it might increase and go north. Now, he also looked at individual species like Douglas fir and ponderosa pine. So this gives you an idea of just those species and what their distribution is expected to be. And again, drought was, or soil moisture was the major control, as well as springtime freezing temperatures and temperature optimum in summer. And Douglas fir doesn't change very much. It doesn't have much predicted expansion. I'm also going to show a series of slides here. But ponderosa pine changes dramatically on the right. And my flux site is no longer ponderosa pine there. It looks like it'll be juniper woodlands or probably sagebrush. So another study that we did, there's this program called the OC lands, or Oregon, California lands, BLM lands. And there's discussion of changing the management on those lands, partly because there have been battles forever for protecting old forests in our region. And one of the bones to throw those who need to, BLM actually is committed to, by law, providing about half of their timber gains in money for the local communities, so keeping local logging communities going. And to do that, the Coast Range is very heavily, intensively logged. And the current logging cycle is about 80 years, and they are talking about reducing it to 50 years. And these are trees that can live 600, 800 years old, so really reducing the harvest cycle. They're already doing this. So the plan is for music forests, spare the old forests, and in this case we called old, or what will become old, 50 years and older. And then clear cut all the young forests in that area to continue to provide the timber supply. And then the semi-arid region, where fire return interval is more frequent and drought is more of a concern, we would thin all the forest to reduce crown fire risk and drought stress. So the combination of those two, this is basically using, I didn't mention this, but it's using inventory data, and it's upscaled using Landsat data. So combining those two, it decreased net primary production for about 30% over the 20-year treatment cycle. Now, we didn't go into future climate with this because this is a data-driven analysis. But the point of it is that we need the science at local or regional scales that's believable to be able to help these people, the managers and policymakers, determine what they need to do to mitigate climate change. So our next project, and all these are overlapping projects, but we wanted to look into the future, and we changed to using the CLM model. And at CLM4, and we did sub-regional or sub-plant functional type parameterization on it because we have the data to do that. And so it allows the traits to vary spatially. And so we had some intensive plots, we called them plots that were scattered throughout different forest types where we measured foliar carbon-nitrogen ratios. And we did a lot of other things on those plots, litter, et cetera. But we found the things that made the biggest difference were to provide sub-ecoregion or vegetation type within ecoregion foliar carbon-nitrogen ratios, as well as mortality changes with age, as been predicted, as we can see in the mortality records and inventory data. And then the wood allocation routines. In this model, CLM4, there's a parameter that is wood mass to foliage mass ratio, so we could compute that out of our data. And that greatly improved our ability to predict. We first ran through with default variables, and then we did this. So on the left is comparison of ecoregion mean net primary productivity from inventories, the white bars, compared with the model, the gray bars. On the right, we also did this seasonally with flux data at several flux sites, and this is just the Matolia site for comparison, to see how well we did seasonally. The blue bars are the model, black bars are the eddy flux observations of GPP. And you can see the interannual variability in the data, in the observations, is much greater than that of the model. So there's more work to be done, and we think it's something to do with drought sensitivity, so we're going to be focusing on that. And here is just future climate, nitrogen deposition, and CO2, so no harvest yet. And we ran the model, or forcing data, from 2 climate models that have been downscaled to 8th degree grid, and then 2 RCP scenarios where CO2 increases to either 550 parts per million for the 4.5 or 900 parts per million for 8.5. And net ecosystem production, the mean over 80 years, increased more, as you might expect, for the higher CO2 scenario, the 8.5, in both of those models, compared with current. And then down in the lower part, we wanted to see, well, how did that turn out when you split it out by the wet ecoregions versus the dry ecoregions? And this is over the state of Oregon. And as you might expect, it was the high CO2 scenarios in the wet ecoregions that showed the biggest increase in net ecosystem production. Well, now we bring harvest into this. And the top graph, same runs, but it's multi-model mean, the black line, showing net ecosystem production from 2010 to 2100. So there's a slight increase. It's going to bump up, but a slight increase over that time frame. And we also, using our maps of vulnerability, we salvage logged those areas predicted to burn. So that's that dotted red line. And then we did no harvest, the blue line that's low there, and clear cutting. And we did this over repeat cycles that I had mentioned earlier that are being applied in the region. So I think the biggest difference you can see is for the no harvest scenario. So when we stopped harvesting in 2010 and just let everything grow, I think this is a growth response associated with age effect on net primary production. Okay, same data on the bottom, but cumulative over the simulation period and net ecosystem production change. And then minus the fire emissions there, that didn't change much in spite of the harvest. Wood harvest increasing up to clear cut, the red bars, and then net biome production on the right. So what it ended up doing is the more severe the harvest, the more that decreased net biome production. And the flip of that is the more carbon emissions, CO2 emissions that result from that. So bottom line, from predictions from these various sources, species richness is projected to increase to the north, decrease in Oregon and Washington, particularly in the coastal areas. And the presence of semi-arid ponderosa pine is projected to decrease substantially over the next 70 years, like about 50%. And then net ecosystem production might increase for the high CO2 scenarios, but harvest to mitigate climate and fire doesn't make up for it. It ends up increasing CO2 emissions over that time frame in Oregon. So the question we're still after, and it's nagging us, is where will forests be vulnerable to mortality in the future, and what can we do about it? And improvements needed on both observations and modeling are regional climate projections. So we have now these new CMIP-5 data, and there are new, more physics-oriented approaches to downscaling, and we need the data downscaled from 1 degree, ideally down to 1/8 degree, but 1/4 is probably the best we're going to get. And then we have to do a better job of modeling the mechanisms that lead to mortality. So right now we can't kill trees in the model, and we want to be able to do that. And then we can get to an idea of how it might shift into something else. So I think with there I'll end it. Just the amber light just came on. Thanks for having me. I'm willing to take questions.
I have a question here.
Okay.
How fast the trees can move? That seems to move very fast.
It depends on the species. So we did some work on this on the biscuit fire, and they can come in. They can reach. The seed fall can go about 500 meters for Douglas fir in a year. And then we looked at that and how the seed fall occurs. It's usually over the first 3 years after a big event like this. And then you'll have a long tail of a little bit of seed raining in. So they can't move fast enough. That's the bottom line. They can't pick up their roots and go. So unfortunately, but we still need to try and think about facilitating that. And I've given congressional testimony and said, let's try and improve the connectivity of federal lands with private lands and maintain biodiversity so that those who can move will, and let them shake it out. We're not very good at engineering this. Okay. Thanks, everybody.
Thank you. Our next talk is given by Kimberly Novick. It's going to talk about observations from 2 side-by-side forests.
That way? Great. Is this there? Okay. Well, thanks for the opportunity to speak here today, and thanks to all of you all for being here. I'm going to give a broad overview of some trends that have emerged from 8-year eddy covariance records collected over an adjacent pine and hardwood forest in the southeastern United States. Before I get going, I want to thank and acknowledge my co-authors. At one point, we were all co-located at the Nicholas School of the Environment at Duke University. And the data I'm going to share were collected in the Duke Forest, which is right there in central North Carolina. In the middle of what you can see is a large and important regional terrestrial carbon sink. In this figure, increasingly blue cells represent increasingly large carbon assimilation by the biosphere. The size of the sink reflects large forest biomass in the region, as well as the warm and mesic climate. The climate is changing. Most GCMs expect that in the future we can expect more extreme climate events, as well as a warmer future in the southeastern United States. The region is also experiencing some significant trends in land cover. There's a trend towards more forest cover overall, reflecting agricultural abandonment. As old farm fields are abandoned, they degrade into upland deciduous hardwood forests. There's also a trend towards more fractional cover by planted pines, reflecting shifts in timber markets that favor the production of southern softwood. And the crux of our research focus is really to ask, in what ways will ongoing climate and land use change affect the magnitude and also the variability of this important regional carbon sink moving into the future? And today I'm going to focus on 4 factors or processes. I'm going to look at the role of vegetation type in controlling annual NEE, specifically by comparing NEE in a pine forest and hardwood forest. I'm going to look at an ice storm as well as multiple drought events, and also explore the role of the length of the active season in controlling interannual variability in NEE, noting that, of course, the length of the active season or growing season is strongly controlled by local climate. Our study sites, again, were in the Duke Forest. From 2001 to 2008, we measured eddy covariance fluxes of mass and energy in a rotation-age loblolly pine stand and a nearby deciduous hardwood forest. I should note that our pine site is actually located in an ambient ring of the Duke Free Air CO2 Enrichment, or FACE experiment. In the interest of time, I'm omitting details about data quality control, gap filling, partitioning, etc. I'm happy to entertain any questions you might have if there's time. We did partition NEE into its principal components: gross ecosystem productivity and ecosystem respiration, and the sign convention is such that negative NEE denotes carbon sink. Here's a look at meteorological conditions during the 8-year study period. The color bar up top is Palmer Drought Severity Index, and you can see the sites experienced 2 drought events, severe drought events during the study period. The first was an early season drought that occurred in 2002 primarily, and the second was actually the drought of the century for this region. It was an extreme drought event happening in 2007. These bars here show quarterly deviations for a range of meteorological variables from the study period mean. And you can see that the drought events are characterized by higher than average air temperature, VPD and PAR, and lower than average precipitation and soil moisture content. The sites also experienced an ice storm event in December of 2002, which primarily affected the pine forest via large reductions in leaf area and branch area. So what you look at here is just a simple bar graph showing annual fluxes of NEE and its components in the 2 forests over the 8 study years. I've highlighted the drought years. Pine is shown in green and hardwood is shown as yellow, and these are summary statistics over here. I'm going to use this figure as a springboard from which to investigate some characteristics of the data in more detail. I want to start with some notes about the study period mean magnitudes of NEE and its components. In terms of absolute magnitude, both GEP and ecosystem respiration are higher in the pine forest on average as compared to the hardwood forest, but the difference between these 2 fluxes, which is of course NEE, is fairly similar. Or in other words, the 100-year-old hardwood forest is taking up about as much CO2 from the atmosphere every year as is the younger and faster growing pine. Okay? At about -500 and -600 grams C per meter squared per year, our sites are really beginning to enter the tail end of the distribution of observed NEE. This figure was made by Dennis Baldacchi a couple of years ago using about 600 site years of data from a broad range of global biomes. And you can see sitting here, our sites really are strong carbon sinks. I want to say a couple words about the ice storm event. As you can see, in the year following the ice storm event, NEE was closest to 0 in the pine forest, reflecting primarily a reduction in the absolute magnitude of GEP. I mentioned that the ice storm reduced leaf area index in the pine site, and it did so by about 30%. The attendant change in GEP was not that great, reflecting the fact that needles left on the canopy had greater access to light. After the ice storm event, this is a closed canopy forest. By year 2004, leaf area index had largely recovered, as had GEP. So when it comes to the effect of this ice storm in this pine forest, we could conclude that, yes, it had a significant effect on annual NEE, but this was a very short-lived effect. Next, I want to look more closely at the 2 drought events. Again, the first one occurred in 2002 and the second in 2007. And in order to disentangle drought events from other factors that might be controlling long-term trends in NEE in these forests, we relied on a simple modeling approach, parameterizing a light-dependent model for GEP and a temperature-dependent model for respiration during the using data from the 2nd and 3rd quarters of each of the 2 drought years. Then parameterize that model using data from the same quarters in the years immediately preceding the drought years. So what you're looking at here is really the change in assimilation capacity and respiration capacity in the drought years relative to the years before. And in all of these figures, a positive change would indicate a weaker carbon sink. In the case of GEP, you can see that in both forests, the drought tended to reduce the size of the carbon sink, but the change was more dramatic in the pine site as compared to the hardwood forest, and in particular in the 3rd quarter of 2007, which again was the more extreme drought event. In the case of respiration capacity, the trends are not as general. In any ways, the magnitude of these changes is not so great as for GEP. So for these forests and these drought events, we can conclude that GEP is more sensitive to drought than is ecosystem respiration, I think there's been a couple other talks in this session that have said as much for other sites. And we could also conclude that the pine site is more sensitive to drought than is the hardwood site. Next, I want to talk about sources, the variability in NEE and its primary components over the 8-year study period. If we look at NEE, you can see that the pine site variability for interannal variability in NEE is greater than the hardwood site, which largely reflects really high interannual variability in GEP in the pine site. Now, the extreme climate events, the ice storm and the drought events contribute to some of this variability. But if you look at this figure, you can see that there's quite a bit of flux variability happening in years that are not directly affected by the extreme climate events. Based on some previous work, we thought it might be a good idea to look at phenology and leaf area dynamics in our forests as possible explanatory variables for this interannual variability. The first thing we did is we compared the annual fluxes to leaf area duration, which is a factor that incorporates information about the timing of leaf on and leaf off, as well as maximum LAI achieved during the growing season. We found that leaf area duration was a reasonable predictor for annual fluxes in the hardwood site, but not in the pine site. Or in other words, in the pine site, interannual variability in NEE and its components is decoupled from the leaf area dynamics. So we looked for another index of seasonality drawn directly from the carbon fluxes themselves. So what we did, we fit a spline interpolation through mean daytime GEP for every site year in the study, and then picked a rather arbitrary threshold of -12 micromoles per meter squared per second to delineate between the active season and the dormant season. This is not a perfect metric. We did try some other approaches. They more or less told the same story, and this is certainly a very straightforward approach, if nothing else. In the pine site, you can see, okay, so then we call the length of the active season the number of days in between these two crossings. You can see that in the pine site, there's quite a bit of variability in terms of the date of both the start and the end of the active season. In the hardwood site, the date of the start of the active season is more constrained, but there's quite a bit of variability in the timing of the end of the active season. And it turns out that this variable, the length of the active season, is a good predictor for NEE and GEP in both forest ecosystems. It's also a reasonable predictor for ecosystem respiration in the hardwood site, but not the pine site, which highlights the fact that in the pine site, interannual GEP and ecosystem respiration are not closely coupled. I'm going to talk a little bit more about the pine site. In the pine site, the date marking the start of the active season alone is a pretty reasonable predictor for gross ecosystem productivity, at least in non-drought years. This regression line here was generated using data collected in non-drought years only. I have shown the drought years as circled points on this figure, and you can imagine that were these not drought years, GEP in terms of absolute magnitude would have been higher, and they might have fallen a little bit closer to the regression line. There's many reasons why the extent to which this forest gets off to a strong start or early start in terms of carbon uptake could be a strong factor on total annual NEE and GEP. We know that photosynthetic capacity in this forest is higher early in the season as compared to later in the season. Also growth, at least growth of above ground woody biomass, actually peaks in May in this pine forest. We're also beginning to look at what might be controlling the date of the start of the active season in the pine forest. So what you're looking at here is just the start of the active season in each year as it relates to meteorological conditions experienced in January, February, and March of each year. So this is mean minimum air temperature and mean VPD. You can see that they're both reasonable predictors for the start of the active season. Of course, these variables are correlated, but not perfectly, and both tend to outperform a typical growing degree day metric. So to conclude, when it comes to vegetation type, again, we saw fairly similar NEE in the pine forest and the hardwood forest, but more variable NEE in the pine forest. The ice storms reduced the size of the carbon sink in the pine forest, but only for a short time. The droughts affect in both forests GEP more than ecosystem respiration and affected the pine site more than the hardwood forest. And the length of the active season is a strong control on GEP and NEE in both sites. In the pine site, this variability is largely controlled by the date of the start of the active season. So looking forward, if our results hold for the broader region, we know that we can expect more pine forests in the southeastern U.S. in coming decades. We can expect more ice storms, more droughts, and perhaps a more variable length of the active season. Taken together, this would suggest certainly a more variable regional carbon sink moving forward. The extent to which these factors add up to affect the magnitude of regional NEE into the future is, I think, still an open question. Anyways, there's some pretty important disturbance events that are missing from this table. For example, insects. It was my great hope that the Duke face experiment would end with a southern pine beetle outbreak, but that did not come to pass. So I just want to end there. I want to thank Gabby Couture and Ram Orden for their thoughts and support, some other folks that worked at these sites, and of course the sources of funding.
Thank you. Well, time for 2 questions, if you have.
Yeah, and it's a monospecific stand, and I think that in the hardwood forest, you have many different species, each with their own unique drought response strategies. Actually, Chris Oishi, who's sitting right in front of you, has done some work in that hardwood forest looking at tree water use and its response to drought. So there's just a bit of a buffer built in by the different species in the hardwood forest.
Okay, thank you.
Thank you.
Our next and final talk of this afternoon is given by my co-chair, Dr. Xiao. He's going to give us very exciting stuff on synthesis of flux tower observations. Thanks.
Okay, here we go. Thank you for staying to the last talk of this afternoon. A number of previous speakers have talked about the impacts of extreme climate events and the disturbances using field observations and ecosystem modeling. And here I included my own presentation at the end of the 2 sessions to provide a slightly different perspective, which is from the upscaling of the FluxNet observations at the global scale. As we all know, as a large number of adequate various flux sites over the globe, taken together, they encompass a large range of ecosystem and climate types. And these flux observations represent the fluxes at the scale of the tower footprint. Over the last few years, we have been using empirical data-driven approaches to upscale the fluxes from the tower footprint to regional, continental, and global scales. And we make use of spatially explicit information, such as a variety of multi-data streams, climate reanalysis data sets, and spatial ecological data. And through upscaling, we estimate carbon fluxes and water fluxes for each grid cell across regions, continents, and the globe. More recently, we upscaled the fluxes from the tower footprint to the global scale and estimated carbon and water fluxes for each 5 kilometer by 5 kilometer grid cell across the globe for each 8-day time interval from 2000 through 2010. This is a global fine resolution data set upscaled from FluxNet observations. What we see here are as the mean annual carbon fluxes and water fluxes. So top 2 panels are gross primary productivity and net ecosystem exchange. So bottom 2 are ecosystem respiration and evapotranspiration. The units for carbon fluxes are grams carbon per square meter per year, and the units for evapotranspiration are millimeter per year. And for NEE here, negative values indicate carbon uptake, and positive values indicate carbon release. In general, these global estimates capture the expected spatial and temporal patterns of carbon and water fluxes. We do a fairly good job for GBP and evapotranspiration. The uncertainty of the net ecosystem exchange is much larger than that of GBP and evapotranspiration. Now let's turn our attention to the interannual variability, which is the focus of our sessions today. And we look at the impacts of disturbances on carbon fluxes using our global flux estimates. And there are some disturbance data sets available for North America. So we overlaid the large fire polygons across Alaska to the anomaly maps of our annual GBP and NEE for a particular year. And we can see there are substantial anomalies in GBP and NEE right within those large fire polygons. And one here, we see similar patterns in the United States, and we see substantial declines in net ecosystem carbon uptake right within the polygon of a large fire, which is a biscuit fire in Oregon. And previous studies have shown that the Hurricane Katrina had a significant impact on the regional carbon budget in the Gulf Coast region. So we overlaid the isotouches of the Hurricane Katrina onto the anomaly maps of our annual NEE. And we see substantial declines in the net carbon uptake in the area that was severely affected by the Hurricane Katrina. And now let's look at the impacts of extreme climate events, particularly severe and extended droughts. Let's start with the year of 2002. 2002 was a very dry year globally. And what we see here are the anomalies of annual GBP, NEE, and evapotranspiration. I also included the palm drought severity index here for comparison purposes. So many regions across the globe experienced a severe drought, including many parts in North America, the Amazon, some regions in Africa, Asia, and Australia as well. And our global evapotranspiration estimates show the evapotranspiration declined in most of those regions affected by the severe drought. And concurrent with the changes in evapotranspiration, there are large declines in gross primary productivity and net ecosystem carbon uptake in many of those regions affected by the drought in 2002. Now let's move on to the year of 2005. 2005 was a particularly dry year. If we look at the palm drought severity index at the bottom here, pretty much the entire southern hemisphere was influenced by this severe drought. And this is confirmed by our global evapotranspiration estimates. Pretty much everywhere in the southern hemisphere showed declines in evapotranspiration in the year of 2005. And this substantial water stress in the southern hemisphere led to decline in gross primary productivity, and which in turn reduced the net ecosystem carbon uptake in those terrestrial ecosystems. Well, in the year of 2010, we also see similar patterns in terms of evapotranspiration and carbon fluxes. Now let's look at the integrated fluxes at the global scale and for each continent separately. And globally, we estimate the annual GBP around 122 to 125 petagrams of carbon per year. And the global annual GBP is relatively stable and it does not vary too much from one year to another, except the year of 2005. In 2005, there was a substantial decline in global annual GBP. And we also see a substantial decline in annual GBP in South America and as well as Africa. And while for other continents, for example, North America, there is some interannual variability, and the maximum difference among years is up to 1 petagrams of carbon per year. And let's look at the anomaly, the annual NE at the global scale and the continental scale. Surprisingly, there appears to be an upward trend in the global annual NEE. And again, the negative values here indicate net carbon uptake, and the positive values here indicate carbon release. So an upward trend here would indicate a declining trend in net carbon uptake. And there is also a similar trend here for South America. For some other continents, such as North America, there is some interannual variability for the annual NEE. And we estimate a global annual evapotranspiration around 65,000 cubic kilometers. And the global evapotranspiration is also relatively stable, except the year of 2005. And we also see a substantial decline in evapotranspiration in South America in the year of 2005, as well as Africa. And the annual variability for annual evapotranspiration for other continents is not as large as at the global scale or South America. So now let us look at these trends and relationships from a statistical point of view. And if we exclude the year of 2005, there is a statistically significant declining trend in global in annual GDP for South America. And the apparent upward trend here for South America is also significant for the annual AEE. And this indicates there is a declining trend for a net ecosystem carbon uptake in South America, which is most likely driven by the changes in the Amazon. And the trend for the evapotranspiration in South America is also significant if we exclude the year of 2005. And apparently was a joint trend in South America over the last 11-year period of time. And the carbon and water cycles are closely coupled with each other. So let's look at the relationships of evapotranspiration with gross primary productivity and the net ecosystem exchange. There is a very strong relationship between evapotranspiration and gross primary productivity, and there is also a strong relationship between evapotranspiration and the net ecosystem exchange. Our results show that this is a joint trend in South America over the last 10 or 11-year period of time, and this joint trend reduced gross primary productivity, and then which in turn led to decline in net ecosystem exchange. And the decline in net ecosystem exchange in South America is so large that there is also a declining trend in net ecosystem carbon uptake at the global scale. Well, in summary, while gridded carbon fluxes upscaled from FluxNet observations are useful for examining the interannual variability of carbon fluxes. So global land carbon fluxes do exhibit large interannual variability, and the magnitude of the variation varies over space. Our results show that extreme climate events and disturbances, particularly severe and extended droughts and the disturbances, are the two dominant sources of the interannual variability of the global land carbon fluxes. Our results show that there is a joint trend in South America over the last 11-year period of time, and this declining trend, this joint trend reduced gross primary productivity and led to a declining trend in net ecosystem carbon uptake. The declining trend in global net carbon uptake is mainly driven by reduced carbon uptake in South America. Our graded flux estimates derived from the FluxNet observations have been used by several research groups to evaluate the simulations of a bunch of ecosystem models at the continental scale for North America, and the results from a water-centric ecosystem model at the watershed level for the US, and results from a boundary layer model for flux sites across the US, and as a result from atmospheric inverse model at the continental scale for North America. And we are currently extending our global fine resolution estimates to December 2012. And so if you are interested in this data set for model evaluations, parameterization, or in the comparisons, please feel free to let me know. I would like to acknowledge the sources of funding. Our work is mainly funded by NSF through our Macrosystems biology project, and NASA through the terrestrial ecology program and carbon monitoring system. With that, I conclude my presentation.
We have time for a couple questions. Jim.
Yes. Is it true now that your simulations show recently over the last few years that global NEE is about 0 to 1 petagram source
Not really. What we show here is are the anomalies of annual NEE. And the reason is partly because through the empirical data-driven upscaling approach, we do substantially overestimate the magnitude of net ecosystem exchange. So in this case, well, it makes more sense to look at the anomalies of annual NEE. Yes, Jim. Well, as I mentioned in my talk, and this declining trend is mainly driven by the decreases in net carbon uptake in South America, perhaps most likely driven by the changes in the Amazon. And according to our estimates, there is a drying trend in South America. And of course, well, these trends remain to be confirmed by independent results.
Okay, final question.
Have you checked to see whether the anomalies you see from the Terra data sets you shown here are consistent with the aqua data or from, say, race estimates and freshwater water storage?
Not yet. So we've recently finished this kind of global scale analysis in terms of intent variability. But that's something we could explore in the near future.
Okay, thank you. And please join me to give another round of applause to all the people who made beautiful presentations.